Okay, so for our final example of how neurons can be hooked up into circuits or arrangements that actually make useful computations, we'll take an example from the visual system. You've seen this picture before. This is the underside of the brain. The front of the brain is up here. The optic chiasm is right here, and so optic nerves coming from the eyes are going to cross here. There is a synapse in the thalamus. Cells in the thalamus send their axons, all these white things here are axons going up to visual cortex. So this is the primary visual cortex in the back of the brain. This is the first cortical place that is going to be processing visual information. Now, some famous experiments were carried out in history. They won some people some Nobel Prizes in physiology. And the basic idea was to simply record from individual cells in primary visual cortex. So you stick these little microelectrodes into the brain back there and record what sorts of visual stimuli will activate one particular cell in the visual cortex. So again, in this diagram we see just the flow of visual information from the retina down the optic nerve. Action potentials are racing down here. There's a synapse in the thalamus. Cells in the thalamus relay action potentials back to V1. By the way, MT is a place where motion is analyzed, and V for color. Alright, what did they find? What they found was that many V1 neurons were responsive to edges of different orientations. And the explanation for this involves understanding how V1 neurons are getting their inputs. Now, on the retina, retinal cells and the optic nerve axons, those cells that are sending action potentials down the optic nerve, they respond to little spots of light on the retina. So at the level of the retina, cells are responding to just whether there's a little spot of light at that region of the retina or not. But now imagine nearby regions of the retina are sending visual information through the lateral geniculate, but that these LGN cells, the thalamus cells, are converging on a V1 neuron. So if there's a little spot of light at this position on the retina, this cell will be active and it'll send action potentials to the V1 neuron. But now again we invoke the spatial summation rule. Let's imagine the V1 neuron can only respond if all three of its inputs are simultaneously active. What that would mean is that you'd have to be shining a little spot of light at this location on the retina, and then the nearby location, and the next location here. Then you would get simultaneous activity in the thalamus cells, action potentials racing down simultaneously, releasing transmitter enough to reach threshold, the V1 neuron response. Well, if you think about three spots of light right next to each other, that's an edge, right? So in the next record here, you see what happens if you put like a bar of light at that location on the retina. You're activating all three of these thalamus cells, and they're converging on one V1 neuron, and you get high frequency action potentials. So this cell is responsive to a vertical edge, in a sense. But notice what happens while you're still recording from that neuron. What if you change the orientation of the stimulating light? So now, notice the bar here might be activating this region of the retina, but not the neighboring ones. So you'll get activity in this center thalamus cell, but these cells will be silent. And the idea is, even though this one's active, action potential is racing down here, it's not sufficient to drive the cell to threshold. So you get lower frequency, or maybe it is a little bit, but you get a lower frequency action potentials. So this cell then is tuned to a vertical edge. We would call this a vertical edge detector. If light at that location on the retina is at this orientation, that particular cell will not respond as strongly as if the light is in a vertical orientation. So these are called edge detectors, and their discovery, along with a lot of other research from these labs, won Hubel and Wiesel the Nobel Prize in physiology. So here's an example, just another diagram of what's going on here. Here we have the same view of the brain, the pathway of visual information. And what's going on here, we got the retina, we got cells in the retina are sort of responding to light at certain locations on the retina. Each cell responds to like a little spot of light, but 3 neighboring cells then could be simultaneously active if you had a vertical bar of light on the retina at that location. LGN cells will be activated, lateral geniculate nucleus of the thalamus, and if they send their axons up to some cortical cells that then eventually converge on another cell, then this cell will respond to vertical edges. As soon as you start changing the orientation of the bar of light, the activity of that cell will drop off. This diagram gives you a sense of how these things might be wired up. You can imagine then the retina is sort of like a two-dimensional sheet, and the placement of these cells are in a position to detect light falling at these different locations. The thalamus is also like a two-dimensional map of the retina, right? So there's kind of like a one-to-one mapping. Cells in the retina, this would be the optic nerve here, and then the cells in the thalamus. Cells in the thalamus send their axons up to cortex, and some of these cortical cells converge on what we're calling these edge detectors. So for example, this cell here will respond to the vertical visual stimulus, and let's see why that is. We can trace it backwards. It gets 3 inputs. If we trace this red input back to this cell is activated by this lower cell in the thalamus, which is activated by the lower cell in the retina. The second input here, activated by this one, that's the middle cell in the thalamus, middle cell in the retina. Third input here, to the top cell in the thalamus, top cell at the retina. So a vertical bar is going to activate these 3 retinal cells simultaneously, which means these cells will be simultaneously active, which means this one, and this one will be simultaneously active, which means this one gets converging inputs at the same time. You get spatial summation, drives the cell to threshold, it responds. But notice, if the stimulus now is this one, well, that's going to activate this one, and this one, which will activate this one, and this one, which will activate this one, this one, and this one, which will converge on this one. So this cell will be maximally sensitive to a diagonal bar of light. Now, there inevitably were researchers that wanted to see whether you could influence this wiring during development. And so experiments were done with kittens, and the kittens were fashioned with goggles. And the goggles only permitted the kitten to see certain edges of certain orientations. For example, if the kitten were fashioned with goggles that only had horizontal lines in it, right, then what they wanted to see was what would happen in the visual cortex if the kitten's early visual experience were restricted in this way. Well, interestingly, what happened was the kittens, later as they grew up into adult cats, and then they recorded from cells in the cortex, they had a whole lot of horizontal line detecting cells. So many of the cells, more than normal, many of these cells were responsive to horizontal lines. Interestingly, they had very few vertical edge detecting cells, and that showed up in the cat's behavior, because the cat would often run around and bump into the legs of tables and chairs. And so here then we can see the connection between sort of the psychology, you know, what the cat is experiencing, and the biology. The cat had very few vertical line detectors. It's as though the cat had difficulty perceiving the vertical edges in its visual world. And so it would run around and bump into tables and chairs. And so that's a nice illustration then that we can understand sort of outward behavior and perception, at the level of perception, linking it to the biology. That in this case, the cat's early restricted visual experience tuned up its visual cortex in an abnormal way. It had a lot of horizontal detecting cells, horizontal edge detectors, very few vertical edge detectors, and that shows up in its behavior. It can't navigate through an environment with vertical edges. This diagram shows the results of a simple experiment where you just sort of take your recording electrode. So this would be the brain, the visual cortex here, that area there. And so what they did was just inserted this microelectrode deeper and deeper and deeper, all the while recording from cells and recording which edges they were most responsive to. And they just pushed the microelectrode deeper and deeper. And up here you can see the records, and down here of the which orientations the cells seem to be maximally responsive to. And so you get this kind of, this cell responds to that orientation. A little bit down here it's more vertical. A little bit down here that's a little bit fuzzy cell. But you go a little further down here, this cell responds to there. This cell here responds to horizontal edges, and so on. And so there are these huge populations of cells that are responding to different orientations of edges. And if you think about the significance of that, I mean, that is a lot of what we see in the environment. We see objects with boundaries. And so what seems to be happening is the visual cortex is beginning to analyze the visual world by detecting the edges of objects. So the first stage of processing is already sort of breaking down the visual world into these edges. Now, we're not conscious of the activity of these cells. We just see edges. We just see the edge. We're not consciously aware that we've got this army of V1 cells that are just responding to particular edges at a particular location in the visual scene. Alright, what you're about to see are some results from the actual lab of Hubel and Wiesel, and they're going to show us a cell that they were investigating. So they had a cat anesthetized, looking at a screen on which they would project light patterns, sometimes squares, sometimes edges, and they would vary the light stimulation, and they were looking for what visual stimulation would cause maximum activity in one particular neuron. The little clicks you hear are action potentials being generated by the cell that they're recording from. So here it goes. Okay, so what you could see there was that only a certain orientation seemed to be activating that cell. And if you flash light on the whole screen, the cell did not respond, and that has to do with some inhibition. Other neighboring cells get activated and they inhibit that particular V1 cell. So that gets a little more complicated than we're going to get into right now. But you got the idea that only light of a certain orientation would activate that cell. So again, spatial summation not only can help us understand the delay lines in the auditory system, helping us localize sound, but spatial summation is involved in the response properties of these cortical cells in the visual cortex. We have edge detectors that begins the process of visual analysis of our world.